Alright, so in this lecture we're going to talk about lipids. Remember, these are going to be macromolecules, but these are not going to be polymers. Alright, so first off, they're going to be made of carbon, hydrogen, and oxygen, which, remember, those are going to be the same elements that we find in a carbohydrate. However, the ratio of the atoms is going to be different. So in a carbohydrate, the ratio of hydrogen to oxygen was 2 to 1. But in a lipid, the ratio of hydrogen to oxygen is going to be greater than 2 to 1. Lipids are going to be used for energy storage. They can be used to make biological membranes, and they can also be chemical messengers. So we'll take a look at each of those in the next few moments. So for our first example, we're going to be looking at something called triglycerides. So these are going to be used for energy. And triglycerides are going to be made up of 2 parts, a glycerol molecule, and then 3 fatty acid tails. Those fatty acids can be saturated or unsaturated, or we can also have a trans fat, which is where we are going to add hydrogens to an unsaturated fat to make it saturated. Okay, taking a look at different types of fats, we have the unsaturated fats. If you take a look over here, you see that the unsaturated fats have kind of a bent or non-linear shape, and that's going to be due to this double bond that we see here between the carbons. We could have multiple double bonds in this particular setup. We have saturated fats. These are going to be straight chain fats, and notice that they have no double bonds between the carbons and hydrogens. Alright, so next up, we're going to have phospholipids. These are going to be used in building of our cell membranes, our plasma membranes. And these are going to have 3 parts. We're going to have our glycerol, which is also then going to have, instead of 3 fatty acids, 2 fatty acids attached to it, and then a phosphate head. Okay, so taking a look here, we're going to end up with, this is our phosphate head. These are our fatty acid chains, and we're going to end up with 2 layers, or a bilayer, for our cell membranes. So these phosphate heads are going to be polar, which means they're going to end up becoming attracted to the water molecules. So these are called hydrophilic, water-loving, or liking water. Okay, down here we're going to have the non-polar fatty acid tails, and those are going to be hydrophobic, and they tend to move away from the water, hence this double membrane structure. Our third group is going to be called sterols. And for these, one of the characteristics that you need to remember is that they've got 4 carbon rings as part of their molecules. And if we take a look over here, we see several different types of sterol molecules. Many of them you are probably familiar with: cholesterol, testosterone, progesterone as well. And if you take a look here, you can see that they all have this characteristic 4 carbon ring shape. Alright, so here we can kind of take a look and compare all the different types. Remember, these are going to be our triglycerides, unsaturated and saturated fats. These are going to be our phospholipids, which are going to build our biological membranes. And then these are going to be our sterols, and these are going to be used as chemical messengers. Alright, so I mentioned earlier, if a lipid is an organic molecule, remember it's going to contain carbon. It is a macromolecule because it's large. Is it a polymer? And remember that the answer was no, because polymers are going to be made up of repeating subunits called monomers, and these lipids are not made up of repeating units. If we're talking about a triglyceride, you have a glycerol and 3 fatty acids, it's done. We're not adding more to make it larger. If it's a phospholipid, we have a phosphate group attached to the glycerol with 2 fatty acid tails. It's done. We're not going to add more to it to make it a bigger phospholipid. And then for the sterols, the same idea. A molecule is made, it's the 4 carbon rings with the various reactive groups. They are done. We do not keep adding them to each other to make larger ones. So that is why a lipid is going to be a macromolecule, large, but not necessarily a polymer. It's not going to be made up of repeating units. Alright, so let's take a look at how we're going to form a triglyceride, and specifically we're going to be talking about this type of lipid. But remember that if we're making a phospholipid, we would use the same process. And taking a look here, we've got dehydration synthesis. We've got our glycerol molecule, which has the formula C3H8O3. We've got our 3 fatty acid chains here. In this case, they're all going to be saturated. And in order to get them to bond together, we're going to be removing water. And in this case, it's 3 waters, 1, 2, 3, by that process of dehydration synthesis. This is going to form something called an ester bond. Okay, and then over here, we end up with our triglyceride molecule, tri meaning 3, 1, 2, 3 fatty acid tails attached to our glycerol molecule. Remember, these bonds are called ester bonds when we are building these triglycerides. So let's take a look and do it step by step. So here is our molecule of glycerol. Here we have 1 straight chain fatty acid. This is going to be our saturated fat. We have another saturated fat here, another straight chain. And then here we've thrown in unsaturated fat. And remember that this is going to be because of this double bond. They do not need to be the same length. Okay, this was just for illustration purposes. All right, so next we're going to look at this special group here. This is called a carboxyl group. It's going to be a carbon double bonded to an oxygen and then bonded to an OH group. And notice that each of these molecules has that carboxyl group. Here again we highlight the double bond between the carbons, and what this means basically is that these carbons are not holding all the hydrogens that they could because of this double bond. We could have extra hydrogens, hence the word unsaturated. Alright, so the next step we're going to do is we're going to look at water being formed from these 3 bonds here. Remember, these are all going to be called ester bonds. Alright, and what's going to happen is we've got this ester bond here. Remember, we removed an OH from one and an H from the other. OH from one, H from the other, OH from one, H from the other. That's 3 molecules of water. This is how we're going to form a triglyceride, but this is also how we could form a phospholipid. Difference being, instead of having this fatty acid tail here, we would have a phosphate group added, and that's what's going to give us the name phospholipid. Alright, so if you guys have any questions on any of this material, make sure that you take notes and ask questions in lecture, and I will see you soon.